I'll get rid of this thing. Okay, well, thank you very much for showing up to hear my talk, and I'd really like to especially thank Mohammed for what has already been a really fantastic conference. The work I'm going to describe today was done entirely by two terrific graduate students, Richard Wang, who's a current graduate student in the lab, and Beth Dumont, who is a former graduate student, who's now a postdoc with Evan Eichler at the University of Washington, and who will also be giving a talk this afternoon. Luckily, Beth will be talking about the stuff she's doing now so that her advisor can talk about things that she did before. All right, I think we can all agree that this is one of the more elegant processes. In fact, you know, without recombination, geneticists would be out of work. And so I really, you know, enjoy studying the basic mechanisms of recombination. And I think we all recognize that there are a number of functional roles for the homologous meiotic recombination. Here are two that get mentioned a lot. And from an evolutionary standpoint, recombination shuffles combinations of alleles, creating haplotypic diversity, which can have the impact of increasing the efficiency of natural selection. Perhaps more importantly, in many species, the proper segregation of chromosomes during the first phase of meiosis requires at least one crossover per homolog pair. Now, despite this functional constraint, what we know is that recombination rate varies dramatically on a number of different scales. If we look within individuals and measure recombination rate across the genome, as we've heard yesterday, it's non-uniformly distributed. So crossovers happen in a fraction of the DNA sequence, but not in all of it. And this appears as you know, heterogeneity in the form of so-called recombination hotspots that are separated by regions that don't appear to recombine. And it's worth saying that we really don't know that much about how these hotspots are determined. There's been some recent advances in terms of looking at genes like PRDM9, but it's still up in the air. If we look on the broader scale, sort of megabase, the idea is that some people think is that it reflects the average across these, this heterogeneity that's happening at the finer scale. We also know that recombination rate varies among individuals. This is from Alec Jeffrey's work, just measuring recombination rates using sperm genotyping at two known recombination hotspots in humans. And you can see that as you look across 26 men in the same hotspot, there's huge variation in the recombination rate. So there's lots of variation on the individual scale. But the observation that interests me the most is that there's recombination variation, recombination rate variation among species. And we can see this looking across eukaryotes. And I think it's fair to say that we know very little about the causes of this variation, despite its important implications for evolution. Now, as we heard yesterday from Adam Otten, evolution on the fine scale looks rapid. Now, again, I want to point out that this is based on a small number of observations and a small number of taxa, but it seems to be, there's growing evidence for this. In particular, if we look at patterns of linkage disequilibrium across the genome and use local dips in linkage disequilibrium to identify recombination hotspots. In chimpanzees, when you find those hotspots, they often don't coincide with the ones in humans, suggesting that there's rapid turnover, and humans and chimpanzees seem to mostly use different hotspots. Here's where I'd like to make a pitch for continuing to study recombination rate on different scales. The functional constraints that we know about, that is in evolutionary sense, the way that recombination rate is likely to be tied to fitness, although we don't know much about that yet, really suggests that selection targets the broad scale recombination rate. That is, the number of crossovers per chromosome arm per chromosome or per genome. I don't know about any real proposal about or data about the fitness that's conferred by an individual hotspot. So we need to consider these constraints on the broader scale if we want to understand how recombination rate evolves at that scale or on the fine scale. So making that connection is important. Hotspot studies at present just don't have the ability to actually estimate current rates per genome, chromosome, or chromosome arm. So I think it's important to take an integrated approach and study recombination on all these different scales. If you want to measure recombination on the broad scale, there are two general approaches available, two general frameworks. One is to follow the transmission of markers in controlled crosses or pedigrees, and another is to use cytological observation, which is what the approach that we've followed. So if we think about meiosis 1 in mammals, which is the focus of my talk, there are several substages, and they go something like this. In the beginning, we have this important protein SPO11 that is generating double-strand breaks. As we heard in the excellent talk from Doug Bishop yesterday, then DMC1 and RAD51 show up and are involved in resecting these double-strand breaks. And then at some point, we have this interesting protein called MLH1 that shows up, localizes at the sites of crossovers. This is a really exciting piece of biology because it means that we can use MLH1 as a biomarker for recombination itself. And in this schematic, we have a pair of homologous chromosomes that are connected by the proteinaceous structure called the synaptonemal complex. And the MLH1 protein lands here, and the foci, if we can just visualize them, tell us about the rate of recombination. So the general approach that we've taken is to extract spermatocytes from mammalian testis, apply a immunostaining protocol, and then visualize and count autosomal MLH1 foci for about 20 spermatocytes from each individual. And the really exciting part of this to me is, unlike most approaches to estimating the recombination rate, what we're estimating here is the recombination rate in an individual. So that means that we can look at variation among individuals in this important genomic parameter. Here's what the images look like. So here the red is showing staining for a protein from the synaptonemal complex. So it's, it shows kind of each of these represents a pair of homologous chromosomes. And there's a stain for a centromeric protein. And then here are the dots that we're most interested in. So we can just go along and count these for individual spermatocytes, do that for a bunch of spermatocytes, and come up with an average recombination rate across the genome for an individual. And what's really great is that the number of MLH1 foci recapitulates known properties of genetic maps. So you can look at this and see that usually you have one MLH1 focus per chromosome. When you have those that have multiple foci, they tend to be spread apart, so you can see interference. It just seems to be a pretty good measure of recombination rate. So I'm going to use this approach to describe 3 different aspects of the evolution of the genomic recombination rate. First, I'll talk about variation within a single family of rodents. Then move on to mention what we think about the genetics of differences between subspecies of rodents. And then I'll end with some new work about the evolution of this genomic recombination rate across a broader phylogenetic distance across mammals. So in the interest of documenting diversity in the genomic recombination rate, Beth undertook a survey of rodents from the family Muridae. We are most interested in this family because house mice are murids. and they provide a genetically tractable system for understanding the recombination rate variation that we saw. But what Beth did was to look at wild-derived inbred strains, two strains each from 3 subspecies of house mice, and then a series of increasingly distant outgroups. Okay, so just using this MLH1 technology to estimate the rates in these different species, the mound-building mouse, the Algerian mouse, the Ryukyu mouse, everyone's favorite, the Norway rat, meadow voles, and deer mice. So here are the results. On the y-axis, we're showing mean MLH1 foci count across spermatocytes, and on the x-axis is a sort of cartoon of the phylogeny between these different taxa. All of these individuals are house mice. These are individuals from the outgroups, and each point represents an individual. And then just in the interest of showing variation, Beth is showing plus or minus 1 standard deviation in this case, okay? So the main result is that although there is variation within individuals, which is I think an interesting observation in itself, there's plenty of variation between individuals, and particularly between taxa, right? So we can see that different species of rodents from the same family have different genomic recombination rates. And there's a lot of action happening within house mice, which we'll come back to in a second. Now these results allow us to kind of revisit some old debates about the old but still active debates about the nature of the functional constraints on crossing over. People have argued back and forth between whether it's the requirement is 1 crossover per chromosome or 1 crossover per chromosome arm. What Beth found for these taxa is that all of them have at least 1 crossover per chromosome, but not all of them have at least 1 crossover per chromosome arm. In fact, interestingly, deer mice fall far below that requirement. So this is an interesting observation. But if we go back to the sort of zooming in on the pattern in house mice, which I think is actually the most interesting, now this is the same kind of plot, but now we're showing the 2 wild-derived inbred strains from each subspecies. So this is polymorphism within a subspecies, and then here's sort of, you know, divergence between subspecies. And again, these are individual animals, but now Beth is showing plus or minus 2 standard errors. So these are approximate 95% confidence intervals. And this really shows that there's a lot of variation among individuals, but particularly, and there's variation between strains within subspecies, but the strongest kind of pattern, or most striking pattern in terms of variation, is the elevation of genomic recombination rate in the subspecies mus musculus musculus, at least as ascertained by looking at 2 strains, okay? So this is a real expansion of the genetic map. And we were just really excited to see this. One thing I'll just remind you of, if I haven't said it yet, is that these 3 subspecies are very recently diverged. For those population geneticists in the audience, if you look at patterns of genomic variation, there's a lot of incomplete lineage sorting. So the estimates are around 500,000 years ago. So this, you know, all of these strains were raised in a common environment. These are genetic differences, and this is a pretty big change. You know, the same number of chromosomes. There's a lot of interesting evolution going on here. Beth did statistical comparisons that showed that both polymorphism and divergence were statistically significant. So this really raised the question of what causes this huge divergence between the subspecies. And again, luckily, because we decided to focus on these MLH1 focus counts, we could actually do the genetic mapping of this trait. And Beth did this in a standard way. She took the strain with the lowest MLH1 focus count and crossed it to the strain with the highest MLH1 focus count. There's about a 30% difference between these two, which I still think is huge. And created F1s, and then recombination in the F1s shuffles the genotypes in the F2s, and she measured MLH1 focus counts in about 20 spermatocytes from each of 276 F2 males. So this is a pretty heroic effort, and I hope that Beth can now go to sleep at night without seeing these green dots in her in front of her, but because she spent so much time looking at these images. So the design is very simple. She counted MLH1 foci and 20 spermatocytes from each male. We genotyped the males across the genome at a set of informative markers, and then you can just do QTL mapping like you would for any other quantitative genetic trait. Our first indication of the pattern of genetic variation was to look at the phenotypic distribution among the F2s. So these are, again, the mean number of MLH1 foci for each F2 male. Here's the mean of the musculus parent strain. Here's the mean of the castaneous parent strain. We have 276 dots here. And the obvious pattern that jumps out is there's a smooth continuum, right? So this is very reminiscent of most complex traits that when you do crosses and map them. This looks like body size, for example. And this is our first indication that there are multiple differences between these strains that contribute to the genomic recombination rate. The broad sense heritability was pretty high, indicating a strong genetic component to this variation. So Beth moved on to actually identify QTL, and we were really excited about this. Actually, we still are. And they had the autosomal QTL that she found had a number of interesting properties. They explained individually about 2 to 8% of the variance in the total MLH1 focus count. Most of the effects were additive. As expected, at these 6 QTL, the musculus allele, the one that has descended from the parent with the high recombination rate, served to increase the recombination rate in the F2s, as you would expect. But one of the most interesting observations here is a negative result, which is that none of these QTL overlap with PRDM9, the gene that everyone seems to be obsessed with, of course, because it's important. PRDM9 is really important in the determination of recombination hotspots. And there's also no overlap with RNF212, which is a transacting factor that affects the genome-wide recombination rate in humans. So we were really interested to see no overlap there. But the QTL that kind of jumped off the page was on the X chromosome. It seems like everything I study in my life, no matter how hard I try to run away from the X chromosome, it keeps bringing me back. And it seems like it's kind of important. What Beth found is that there were a couple of QTL on the X chromosome, but in particular, this huge one that explained a whopping 35% of the variance in the genomic recombination rate. But it got even more interesting when Beth looked at the effects of this QTL, where the allele from the high parent that, remember, increases the recombination rate, the allele from the high parent actually decreased recombination rate, okay? So we have this interesting sort of flipping of the effects that looks completely different from the inheritance mechanisms that Beth identified on the autosomes. So we can think of the kind of distribution of variation by looking at the effects of these QTL in sort of a heuristic way. So there's about 8 foci that separate. Think about 8 foci, right? It's like 4 extra chromosomes. I mean, it's a huge amount of recombination. So that separate these two representatives of these 2 subspecies. If you just add up the effects of the autosomes, you go a long way towards explaining that difference, which statistically you can't really do this. But it gives you an indication of our power to explain things. And then you have the X chromosome effect sort of pushing back in the other direction. So at the end of the day, we're explaining quite a bit of the variation but there's real genetic complexity going on here. This observation of an X chromosome effect that was not what we expected really prompted Beth to work hard to measure recombination rates in females. The reason that we focused on males from the beginning is that this is extremely difficult because recombination happens in the fetal ovary. So, but Beth was really determined to do this. She tried this many different times and eventually got it, and it was, I think, it turned out to be a pretty important result. If you look, remember this is the low parent and the high parent from males, and so you see that that's still true, that the recombination rate in this musculus strain is higher than the one in the castaneous strain for females, but the difference is very small by comparison to what we saw in males. And in particular, if we put males back on the chart, what you can see is not only does the male difference dwarf what's seen in females, but the pattern of sexual dimorphism is flipped, which again, it seems like a pretty exciting result in light of this, transgressive effect on the X chromosome. How much time do I have, Mr. Moderator? I'm sorry, Dr. Moderator. 8 minutes. Okay, good. Thank you. Okay, so I'd just like to finish up by, now we've kind of taken a look at short time spans in terms of evolution, and now I'd like to zoom back out and talk a little bit about some new work done by Richard Wang to look at the evolution across mammals. And the basic idea here, which has become sort of a big part of my research program, is to, again, just think about the genome-wide recombination rate as a complex genetic trait. And we can analyze it in many ways the same way we would analyze or treat other complex traits, other quantitative traits. So to do this, what Richard did was to collect average MLH1 focus counts from every mammal that we could find where people have done this. And Pavel Borodin, who's an MLH1 expert and just loves to catch things, kill them, and estimate their focus counts, provided a whole, rodents, sent us a bunch of data from a whole suite of voles. And in total, what we have here is 31 species. So it's a relatively small size for this kind of analysis, but we're just getting started on it. And we actually have collaborations going to extend the number of species quite dramatically, we hope, across the mammalian tree. And so what he did was he took a molecular phylogeny that was published a few years ago for mammals that was based on a large number of loci and a very large number of species, and just took out the subset of species that we had data for, and then analyze the phylogenetic distribution of MLH1 focus counts using an approach called Bayes traits. And what you need to know about this approach really is that like pretty much all phylogenetic comparative methods, it models trait evolution as a Brownian motion. So you have sort of random fluctuations up and down throughout the tree, and the distance in terms, or the difference in the number of MLH1 foci for a pair of species is just a function of the total divergence time that they're separated by on the tree. So here are the 31 species, and I just wanted to point out, this is hardly a random sample of the mammalian phylogeny, but I think it's still useful to get started. So we have an insectivore, some carnivores, some artiodactyls and a perissodactyl, a few primates, a bunch of rodents. Here are all these voles. And I'll just point out, for those of you who care, that still all of these rodents, despite sampling quite a bit of diversity here, they're all from one family. They're all murids. So what we're working on now is including other things like squirrels, and to really kind of expand this. But the range of MLH1 foci across the tree was about 16 to 50, so that's a huge range. But you will all recognize that these species differ in the number of chromosomes. So once you divide by the number of chromosomes, it's much less. Still, we have a range of 1 to 2.5 foci per chromosome. So the question is, how are these rates distributed along this tree, and can we draw any conclusions about evolution from that? So the first result is looking at something called lambda, which is a parameter that measures the degree to which the variance in recombination rate is predicted by the shape of the phylogeny. So in the case where we have a lambda value of 0, it means that there's no relationship between the shape of the tree and the distribution of recombination rates. When lambda is estimated to be 1, it suggests that there's a very, very tight relationship and that the Brownian motion model fits very well. And so if we look at the number of foci, the number of foci per chromosome, the number of foci per arm, the number of excess foci relative to the number of arms, all of these have, even with 31 species, strongly significant values of lambda. So these stars are based on likelihood ratio tests against the null that lambda is 0. And you can see that the values are relatively high. So there's a clear relationship between the distribution of recombination rates and the tree. And lest you are concerned that this is just reflecting changes in things like chromosome number, I can tell you that it's not. At least in this subset of species, that trait of chromosome number is not tracking the phylogeny, which is kind of an interesting observation in itself. I'm not sure that that's going to be true in general. This is just a sample of 31 species, but anyway, in light of that, we can confidently say that what we're looking at here is evolution of the recombination rate and not evolution of chromosome number. We can look at another parameter here, and I'll just finish up by just quickly describing this analysis, called delta. And the basic idea here is that delta describes the transformation of the phylogeny that is necessary to fit the phenotypic data. So for example, if we look at a delta value of 1, the branch lengths, the ratio of external to internal branches, is the same. It's identical to that in the real phylogeny, the molecular phylogeny, okay? Yeah, alternatively, if we look at delta values less than 1, what you see is a relative expansion of internal branches. So the internal branch length is much longer. The topological relationships here are not changing, it's just the branch lengths, okay? And this is often referred to, because internal branches come first temporally, as a slowdown in the evolutionary rate throughout the tree, okay? So these external branches are shorter, and as you get closer to the present, the trait variation, the evolution of the trait is slowing down. And we can flip that on its head and look at values of delta that are above 1, and in this case you have an expansion, a relative expansion of external branches, and this is referred to in the phylogenetic comparative literature as a rate speed up. So we can, just ask with our data, the MLH1 focus counts, which situation it corresponds to. So here's the molecular tree again. Here are the branch lengths. This is just based on DNA. When we do the analysis and find the maximum likelihood estimate of D, it comes out at 2. So it's greater than 1, in case you didn't know. And what this shows, you can see, graphically that there's an expansion of the external branch length. And again, this is consistent with the idea that recombination rate has been, the evolutionary rate of divergence in recombination rate has been speeding up as we move towards the present, at least in a subset of taxa, okay? So the goal here is really to just begin to get some insights into what's driving, evolutionary divergence in the recombination rate. And this is a preliminary result, but I still think it's encouraging. Okay, so I'll move on to summarize the results here. In terms of the evolution of Murid rodents, one of the factors that I didn't mention is before Beth did her work, the sort of paltry number of taxa in which we had measured recombination rate had raised the idea, the possibility that the low recombination rate in house mice might be a general property of the rodent lineage. You know, people were sort of wondering if that was true. And it turns out that at least looking at it across a family, the largest family of rodents, that low rate is not restricted to house mice. So that's interesting. Beth found, you know, at least one crossover per chromosome in all taxa, but not one crossover per chromosome arm in a couple of those taxa, which I think is an interesting observation. There was lots of variation on different scales, including rather dramatic differences between subspecies of house mice. The genetics of these subspecies differences looks to be pretty interesting, with a number of autosomal QTL that have additive effects, an excellent QTL showing something different entirely. I should point out that we know, while in general we can't say much about whether these QTL are cis or transacting, there is one exception, which is that Beth measured autosomal focus counts, and the QTL with the largest effect is on the X. So that's definitely a trans recombination modifier. Is that? She also found that there's no overlap with genes known to shape variation of the trait, reversal of sexual dimorphism, and then we have this phylogenetic signal and a pattern consistent with an evolutionary acceleration to the present. So the implications are that there's been a potential rate reduction along the rodent lineage, which I think, you know, nominates rodents as an especially interested taxon to look at this in. The chromosome number seems to be functionally more important than arm number. I would say most of the genes our genetic results show remain undiscovered, and we need to be cautious about assuming that PRDM9 explains everything, because it definitely doesn't. I love saying that. It just feels good inside. And then I think we can also say that, you know, different genes affect the fine and broad scale rates. There is the potential for conflicting selection pressures on the sexes. And this is an early, very preliminary indication from our phylogenetic analysis that recombination rate on the genomic scale may be a target of directional selection in at least a subset of the taxa we've surveyed, and we're in the process of investigating that further. Thank you.